So welcome everybody to our Solve-RD webinar on the RD-Connect Genome-Phenome analysis platform GPAP. Today we have our first webinar of a series of 2 webinars in total, and we will learn about general analysis features of the platform. As long as people are still joining, I would like to start with some general housekeeping notes concerning the format of the webinars. We are using the Zoom webinar platform, and you as attendees can use the chat function at the bottom of your Zoom control panel to post to the panelists or to all other attendees. For questions to the speaker, please use the Q&A function that you also find at the bottom of the Zoom control panel. In the Q&A panel, you can also see the questions of other attendees and can like or upvote them. This will help us to pick the most relevant questions for the discussion after the presentation. Now I would shortly like to introduce the panel of our webinar today. The webinar will be given by Stephen Laurie, who is a lead data analyst at the CNVs in Barcelona in Spain, where he has been involved in the RD-Connect project and the development of the RD-Connect GPAP platform. We are happy to have also Leslie Matalonga in our panel. Leslie is a clinical genomic specialist at CNVs, and she will give you insights into advanced analysis features of the RD-Connect platform in the second webinar. And today, Leslie will help with the discussion. And we have Luca Zalatnay on board. Luca is the RD-Connect help desk coordinator and the person to get in touch if you have any issues when working with the platform. And from the Solve-RD Project Management Office in Tübingen, in Germany, it is Birte Zurek and myself present. Now some very brief notes about the project Solve-RD, solving the unsolved rare diseases. Solve-RD is a project funded by the European Commission for 5 years. It involves partners from 10 countries and is coordinated by Olaf Ries and Holm Kressner in Tübingen. Solve-RD makes use of existing rare disease expertise and infrastructure as RD-Connect, EGA, OrphaNet, to name only some of them. This slide now quite impressively highlights the different partners throughout Europe that contribute to Solve-RD. The project is built on a core network of partners from mainly 6 European reference networks for rare diseases and 2 national undiagnosed disease programs. The main aim of Solve-RD is to solve large numbers of rare diseases for which a molecular cause is not known yet. For that, Solve-RD follows 3 main approaches. First, massive data collection and reanalysis. Second, applying sophisticated combined novel omics approaches. And third, by supporting candidate gene validation projects in model organisms. And this last slide now indicates our progress and achievements in the project. We have meanwhile more than 21,000 datasets collected, so even more than originally anticipated. We have the proof of concept that the reanalysis approaches on our first data freeze led to a diagnosis in more than 9% of the included families. And with more than 2,000 novel omic samples being processed and new data being generated, we are convinced that the SolveRD project can further increase the diagnostic yield for many previously unsolved rare diseases. And with keeping this in mind, I would now like to hand over to Steve to give you insights on how data sharing and analysis via the RD-Connect GPAP platform works.
Okay, thank you very much, Cornelia, for the introduction. Okay, so my idea today is maybe to take up about 35 minutes, I think. And then we'll have some questions, 15 to 20 minutes available for questions at the end. So you can fill in the questions in the chat box as we go along, as you go along. So I'm going to start with a brief presentation that will take 5 to 10 minutes, and then I'm going to show you a demonstration of the GPAP in real time. So thank you to everybody for joining today. So I'm going to show you how to solve rare disease cases with the GPAP. So first, a little bit of background. So the GPAP was originally developed within the RD-Connect project, which was a 6-year EU framework program project. And the objective of the project was to generate an integrated platform connecting genomic data, clinical information, registries, and biobanks for rare disease research. Essentially, what we were trying to do is overcome a number of barriers. Do you see the whole screen here, or is it cut? Can you see the pink circle on the right-hand side, Cornelia?
We can see there is like a square, but
yeah, okay,
we can see the whole slide. Also, Stevie, if you can speak a little bit louder, I think some people were having troubles with the sound. Thank you.
Okay. So, so the practical objective of the project was to overcome a number of bottlenecks that hamper rare disease research. So these bottlenecks are in terms of issues of privacy, infrastructure, lack of standards for sharing, a reluctance to share in the community, and a lack of capacity to analyze the large amounts of data. And while the RD-Connect project itself finished in 2018, ongoing GPAP development has been supported through a number of sources, not least including the Horizon 2020 SolveRD project and the European Joint Project for Rare Disease. So what is the platform? So as we like to call it, the GPAP. So essentially, just looking at the figure on the right here, the idea of the GPAP is that it's a scalable and interoperable online system that facilitates collation, sharing, analysis, and interpretation of integrated genome-phenome datasets. And today we're very much going to be focusing on this analysis and interpretation of the datasets. So if you start off as the clinician and the researcher on the left here, the first thing you do when you join up with RD-Connect is that you would provide a phenotypic description of your patients, and that would be described in terms of HPO terms, which go into our PhenoStore database, and I'll come back and explain a bit more about this. And at the same time, you give us, you upload your raw data. We have an Aspera client for this, so this is your sequencing data, and a little bit of metadata so that we know what type of data it is and who it belongs to. And then we take this all together and we put it into the RD-Connect GPAP. And once it's there, this allows the clinician or the researcher, the submitter of the data, or someone in their team to actually undertake their own analyses within the platform to try to solve the rare disease, with the objective of solving the rare disease cases themselves. And also we have a connection to the EGA so that if the data is to be archived for long-term usage in the future or long-term access in the future, we can push the data to the EGA, which is the European Genome-Phenome Archive. So this is the first step, is the phenotypic data. So this is the PhenoStore module, which we built fully in-house in CNAG in Barcelona. And the PhenoStore is a user-friendly tool that enables the collection, sharing, and exporting of standardized phenotypic information, so just the phenotypic information. And the standards we apply are HPO from the Human Phenotype Ontology, ORDO, Rare Disease Ontology, the Orphanet Rare Disease Ontology, and OMIM. And it's an interoperable system, so this means it's connected through APIs, application programming interfaces, which means it can communicate directly with other systems, and in this case, particularly it communicates with the genomic analysis side of the platform, but it can also communicate externally if required. And this was developed in-house, as I say, by Tassos, Alberto, and Davide Piscia, who leads the team. So this is what the top half of a PhenoStore record looks like, if you like. So we have a little bit of a description, the IDs on the left, and you can see this is actually a trio, right? So we have the child who's colored orange at the bottom, because they're affected, and then they have 2 unaffected parents. And then at the bottom, you can see the key bit here. You can see a number of things, but the key bit here is the HPO terms that describe the patient, right? So in this case, patient has Pfizer hypertonia, neck muscle weakness, arthrogryposis, multiplex congenita, and doesn't have, so this is a symptom that's not observed that could be interesting for the diagnosis. This individual, whilst they have all these neuromuscular issues, they don't have any cognitive impairment. To move on to the genomics analysis module, which is what I'm going to be mainly demonstrating today, it has 3 sections. So at the top of the page, essentially, you have to define the samples that you wish to analyze, naturally. Then in the second section here, we have the filters you wish to apply. So, a whole genome has somewhere between 3 and 4 million variants initially. And we're trying to get down to 1 or 2 variants that might be responsible, or hopefully to identify the 1 or 2 variants that are responsible for the rare disease. So you want to apply a number of filters of different classes to try to get down to this small number, and you'll see them as I go through the demonstration. And then once you apply your filters, you run the query, and you will get your results at the bottom half of the page, or the bottom third of the page. So this is just an example of one of the filters menus. So this is the variant type filter menu. And you can see here by clicking in the checkboxes, you can select the type of variant you're looking for, high impact, moderate impact. You can check if it's in ClinVar as a pathogenic or a likely pathogenic or VUS, etc. And you can restrict to just protein coding variants or variants that are tagged within the platform, which I won't be talking about today, but Leslie will explain about the tagging in the further, more advanced webinar in 2 weeks' time. And then we have these other filters, population allele frequencies, effect prediction tools, filtering by genes and disorders and phenotypes, and filtering to particular regions of the genome. And this is what the results look like. So at the bottom half, we have 1 line per variant that's discovered that matches all the filters, a bit of color coding to indicate which variants might be worth looking at first. And then above this, we have an extended menu of results, which for each specific variant, you have a number of tabs that will give you more information about that variant. A useful feature that we've incorporated, one of the many, is that if you click on the coordinate on the left-hand side, or you click on the gene name in the middle here, you get a drop-down menu that provides you with hyperlinks directly to this position in the appropriate database. So if you click on Ensemble here, it will take you exactly to position 233,349,186 on chromosome 2 in Ensemble or Exact or Nomad, etc. And the same for the gene. If you click on ECL1, if you click on OMIM, sorry, if ECL1 has an entry in OMIM, it will take you directly to that entry, or it will take you to that entry in the HGMD or in ClinVar, etc. So the idea of the platform in general is it makes this task of going from 3 million potential variants, or 100,000 maybe if you're doing exome sequencing, to the 1 or 2 that cause the disease. And so who is using the RD-Connect GPAP to date? Well, at the moment, we have over 600 registered users. We have shared data from over 100 research groups and hospitals across Europe and some further afield. And currently we have 20,000 Genome-Phenome data sets. So that's the genomic data matched with the phenomic description processed and uploaded into the GPAP. And this year, currently, we're increasing by approximately 1,000 data sets per month. Okay. So now I'm going to go to what we call the playground instance. So let me stop sharing this window. And let me share another window. Okay. So what you're seeing now is the RD-Connect homepage. And what I'm going to use, as I say, is something we call the playground version. So I'm going to click out and try out the GPAP modules. And essentially what we have here is we have an open version of the platform that doesn't have any real patient data in it, of course, but has basically all the functionalities that you will find in the real platform once you become, if you join RD-Connect, so it has the full functionality. And you have access to the genomic analysis module, which is what I'm focusing on today, but you also have access to the PhenoStore module to see how that would work. So, as I say, I'm going to log into the analysis module. All this information it will be open to you after the webinar today, so you can try it yourself. And the information is provided for the playground. The username is test and the password is 1234. Okay. So this is the screen that you'll see first when you log in. Okay, and as I said, the first thing we want to do is we want to select the samples that we wish to analyze. And we're going to analyze so yeah, I should explain what we've got here, but first I'll type it in. So first we have a number of synthetic families we've made, synthetic trios. The number here defines the family. So this is family 1, and C is the case. And naturally the parents are, the father is 1F, one case, family 1 father, and the mother is 1M. Okay. So first, let me explain a little bit more about what we've done here. So what we've done here is we've taken publicly available genomic data from the Illumina Platinum Genome dataset, where they've sequenced the HapMap sample NA1, they've performed whole genome sequencing to 50x coverage of NA12878, which is the most sequenced individual in the world. And also we have her parents here. So NA12889 and 90, I think they are, but it's in the information. So we have a real trio here, but what we've done is we've taken actual causative variants that were found by the GPAP in one of the real cases, and we've spiked them into the VCF file before we have uploaded the variant to the platform, right? So we have a real variant here in the child, and the corresponding variants in the parents, and we're going to try to solve the case on this background of a publicly available genome. Okay. So you can see when I've put this in, the first thing that appeared as I was putting it in is that we have a link to PhenoStore, right? So let's go have a look at this case in PhenoStore. So the login is all the same. So as I'm logged into the GPAP, I'm logged into the analysis, and I'm logged into PhenoStore at the same time. So this is our case. So in PhenoStore, you can see who submitted the case. This is our internal group, obviously. Sergei Beltran is our leader. The family gets an ID, and you can see the samples that have been submitted. So in this case, it's just a simple pedigree, as we have the free PhenoStore IDs here. This means that we have the free records in the system, and we have the corresponding genomic data. This is the record for the child who is affected. And if we go down, we can see, I'm just going to hide the diagnosis. we can see the rest of the record, as I say. So this is similar to the one I showed before. These are the HPO terms that have been associated. Why do we use HPO? Well, the important thing about HPO is that it has a number attached. Every term has a number attached. So neck muscle weakness is HPO number 467, and the inability to walk is HPO number 2540. Why is this valuable? Well, this is valuable because it means computers can talk to other computers, right? So computers don't really understand the inability to walk, but if you send them 0002540, they can check, do they have that number, or do they have a number similar to that? So this is a way of sharing phenotypic data. Okay. So to go back to the platform itself. Oh, sorry. One last thing I should highlight here. So this is as if we had uploaded the case, right? And the mode of inheritance, well, you can see the parents aren't affected. So this is unlikely to be autosomal dominant inheritance. And the mode of inheritance that the submitter actually provided was sporadic. So sporadic in this case means this is the first case known of in this family as far as they are aware, okay? So for the purposes of today's demonstration, at least, I'm going to assume this might be some sort of autosomal recessive case, right? So the parents would be carriers. So they would be heterozygotes. They would have one allele that looks like the reference and one allele that's alternative. And likewise in the mother and the father. So they both have one bad allele, if we like, alternative allele. And then we're expecting the child to have two alternative alleles if it's autosomal recessive. So they're homozygous for an alternative allele in this case. And this may be what's causing the disease. So you can run a query at any point, right? So as I say, in general, we have 3 to 4 million variants per individual that are different from the human reference genome. And if we run the query here, we get returned all the positions where the parents are heterozygous and the child is homozygous at the same position for the alternative allele. And you can see here the number of variants we get back initially. I hope you can see my mouse. I'm not sure if you can see my pointer or not, but the number of variants is displayed here in the results section, and there's 220,000 variants that match this inheritance pattern, right? So on the basis of this is not particularly helpful, right? 220,000 possible explanations. So what can we do? Okay, well, we want to apply filters. So the first filter menu we have here is the variant type menu, right? And if you hover over, and again, I'm not sure this doesn't always show up on the webinars, but I have placed the cursor over variant class, and now it has a little pop-up, a tooltip, which tells us what the variant class is. So the variant class here is as defined by SNP-F. This is actually as now defined by the variant effect predictor by Ensemble, but the terminology is the same, and you can go find out what each means. Likewise, if I hover over high, I find out what high is. And in case you're not seeing it, what it's saying is a high effect variant is a variant that's assumed to have a disruptive impact on the protein. For example, nonsense variants, frame shift inducing variants, and splice affecting variants. So these are things that damage the protein, while moderate impact variants are variants that are predicted to affect the protein, but not necessarily, well, they affect the protein sequence, but they don't necessarily damage the function. Of course, certain non-synonymous variants will have an effect, but just looking at them all, looking at them initially, we can't be sure whether they're likely to have an effect or not. Okay, so usually if we're interested in protein coding variants, and that's usually what we are interested in, we will apply a high and moderate filter. And this, in this case, takes us down from 220,000 to only 620 variants. Okay, so that's quite helpful. 623 is a much better number, but we're not going to be able to go through 623 variants one by one and try to find which one matches our case. So we can carry on and apply further filters. So because we're interested in rare diseases, the big advantage of rare diseases is that they're rare. And this means that the variants have to be relatively rare as well. So a rare disease in Europe is defined by something that affects less than 1 in 2,000 members of the population, right? So we can basically we can use the data from Nomad to filter out common variants, right? So I'm filtering out anything that has a allele frequency of more than 2% here. It's defined as a proportion, so 0.02, but it's 2%. That has been observed at more than 2% in either Nomad or the 1000 Genomes project. Usually I would also use the internal frequency as well, but as these are simulated cases all based upon the same background, the internal frequency isn't useful here because we only have a sample size of 3 that's been repeated 10 times. But we can look for the variants that are at least rare in this trio. So we're looking at 2%. So this means a combined the probability of having 2 alleles by 2 bad alleles would be 1 in 2,500, which is more or less where rare diseases come in. And you can see now we've got down to 33 variants, right? So already we're beginning to reach a useful number, right? 33 is almost a manageable number. If you're an expert in the disease or the class of disease that the child has, excuse me a second. So if you're an expert in the class of disease that the child has, then you may be able to go through these quickly and identify which genes are not going to be of interest and which are going to be of interest. Of course, if the child has something unusual, you're not very sure what genes are likely to be involved. So the next filter that's useful to apply is a filter based upon the genes, right? So we have a number of ways of doing this within the platform. So this is one of the strengths of the platform, is it has a wide variety of different ways of trying to solve your case, because some cases are very difficult to solve and they're not immediately what they appear. So you need to be able to tweak these parameters but get the results back quickly, which is not something that you would be able to do if you were still doing this in the old-fashioned way, which was basically getting some results, a short list of results, a manageable list of results in an Excel table, and then trying to filter by all the different possibilities within your Excel table. So here, what we have is a number of different ways of defining the filtering by genes. So one thing we have is we have a number of built-in gene lists. Actually, the gene lists that are built into the full platform are about twice as many as these, but these have been sourced either from researchers who use the platform a lot and have told us what is useful for them, or from genomics rare disease community gene lists that we all know are useful. For example, we have the Decipher gene list, all genes that are used by Decipher, which was 1,692 back in July 2017 when this was made. We have a number of genes that are known to be imprinted. So this is 253. If you're studying genes that diseases that you know are likely to be imprinted, you could filter to these. In this case, because we have a neuromuscular gene list, a neuromuscular phenotypic description, a useful gene list is this one from Muscle Gene Table in France, which is now this version, as you can see, is 5 years old and only has 416 genes on it. I think the current version has 611 genes on it, so it's very much more extended, and that's what we have in the platform. But today we can select the Muscle Gene Table and see if this has any effects on the 33 variants. So we're only looking for genes that are known to be associated with neuromuscular diseases. And here you can see we get down to 2 cases. So of course, there's always the possibility that the gene hasn't been associated with the particular phenotype this child has, right? But let's have a look. So there's 2 genes that have been involved in neuromuscular diseases that are found in the correct pattern, where the parents are carriers and the child has a homozygous alternative variant, right? So these 2, one on chromosome 14, one on chromosome 22, and the first one's affecting ataxin. So if we don't know anything more about the gene, as I don't, because I'm not really an expert in these things, I can use whichever is my favorite resource, PubMed or Ensemble, to try to find out more information. Of course, one of the most useful resources initially is OMIM. So this is ataxin-free. We go straight to ataxin-free in the OMIM. And the first piece of information that indicates to me that this ataxin 3 is not likely to be, this variant in ataxin 3 is not likely to be disease causing, is because regardless of the phenotype, is that its inheritance model is autosomal dominant. And here we have an autosomal recessive case. So I can close that down immediately, and let's go have a look at NEPH and see what it does. NEPH, according to OMIM, is the neurofilament protein, heavy polypeptide. And you can see that it's involved in two diseases, implicated in two diseases under an autosomal dominant inheritance pattern, but also one disease with an autosomal recessive inheritance pattern. So let's have a little bit more look at this. So the autosomal recessive disease is amyotrophic lateral sclerosis, better known as ALS. This is a susceptibility to ALS, autosomal receptive susceptibility to ALS, also known as Lou Gehrig's disease. And you know, you would have to look through the description here and compare it to the description, but it's very clear if you know a little bit about ALS and we looked at the patient description that this is not likely to be the explanation for what this child has. So if we look at the PhenoStore record, it has a number of features here, a number of HPO terms here that are not associated with ALS. So it looks like here our investigation hasn't been successful. So what can we do? Well, if you remember from the PhenoStore entry that the inheritance pattern is sporadic, right? So there's a couple of other possibilities that would solve a sporadic inheritance pattern. One, and you're probably all screaming it, would be, it could be a compound heterozygous mutation, right? And if you want to search for compound heterozygous, actually it's more likely it would be compound heterozygous and simple autosomal dominant, then you can click this box. And because compound heterozygous inheritance requires that the father carries one allele and the mother carries a different allele and the child carries both those alleles, we gray out the boxes for the inheritance pattern and we apply it directly the compound heterozygous filtering. So I'm going to remove the gene level filtering and apply the compound het filtering. Okay, now, so what we're looking for here is two hits in a gene, at least two, right? Because the child has to carry both alleles in compound heterozygosity. Both alleles have to be rare still, because here I'm still keeping the filters from before, right? So I'm using the same filters as before. You saw I switched off the genes, but I'm applying them directly, and you can see we got the results back essentially instantaneously, or in a few seconds, for the compound heterozygote positions. And there's, you can see we have 47 variants here, so we have at least 23 different genes. And again, you can go through each of them, or we can apply other filters. You see by the color coding which ones might be more interesting. You also have the allele frequency here. So if you happen to know your disease is at a lower frequency, the 1 in 2,500, let's say it's less than 1 in 10,000, then actually you would want to remove all variants here that are above 0.01 in Nomad, because they're too frequent to be causing the disease. So let's look at another way we can do the filtering. So something I haven't shown you yet is that if you click on the phenotype tab here at the bottom, you also get all the HPO terms that are in the phenotypes record, the PhenoStore record. So without having to go through to PhenoStore, you can see the HPO terms that are affected. So what we can do, because everything's linked via APIs, is we can actually fetch the HPOs directly from the PhenoStore record. There's 10, and then we can apply, we can generate a list of genes on the fly, so not using our predefined gene list here, but ask the HPO ontology or Mendelian or DisGenet, can you give me a list of genes that have been associated with these HPO terms that cause rare diseases. What are those genes? And you can see if we use the HPO ontology, we get a very long list of genes. There's about 1,500 here, I think. And that's because, or most likely because, in the PhenoStore record, we have some terms that are very high up in the HPO tree. For example, muscle hypotonia basically means that you've got weak muscles. So any gene that has been associated with a disease that has a phenotype of weak muscles will be showing here. Nevertheless, although we have 1,500 genes or so here, that's still only less than 10% of all the genes. So if we apply this, you can see we're down to 19 variants, a large number in HLA DRB1. So these probably aren't likely to be of interest because they're in the MHC. And you might want to look in more detail at these two at the bottom, NXA11 and RTTM. However, if you did so, you might still not find a disease-causing variant. So what would you do then? If you discount them quickly, then what we can do next is, I'm going to remove the gene list again. So this is the first time this has been seen in this family. So this might be one of these relatively rare cases, but relatively common in rare diseases, where the individual, the case, actually has a de novo mutation in a gene, right? So the father and the mother have a reference-like position, but the child has an actual mutation that results in it being heterozygous, and it has this one heterozygote is enough because it's a de novo autosomal dominant cause of disease. So again, let's run the query. I have to clear the HPO gene list. Okay.
Steve, also select and deselect the ref alt because the allele frequency has not changed. Just a bit. Perfect. Thanks.
Thank you, Leslie. As you can see, Leslie is really our expert in-house user. And well, let's run the query. Okay, so initially we have 56 variants that look like de novos. Many of these would actually be false positives. They'll be in regions where we actually don't map very well the genome, and these are identifiable. For example, they have suspicious looking alternative allele frequencies, but I'm not going to go into that detail here today. But regardless, we're not really interested in the long list because it's still the 56 and we haven't applied our gene list yet, right? So let's get our HPOs again, and let's use the HPO ontology again and run our query. Yeah. And there's one other key thing here I haven't done. Okay, so now we've got 6 variants that match our filters, right? And actually I see the other one. The other thing I could do is reduce the allele frequency in the populations, because if this is a heterozygous case, then the allele frequency will be less than 1 in 2,000, right? So we could go down to 0.0005. But as I see, if you look in Nomad, actually 4 of these variants have never been seen, and the other 2 have been seen at very low frequency. So one probably 1 incidence, and the other one maybe 3 times, something like this. You could go and check in Nomad as well. So you can see by using the HPO, we're down to 6. But let's see if one of the other tools can do us a better job. So we also have a direct connection via API here to this Genet, which is a database, again, developed in Barcelona, but not by ourselves, which scans the literature essentially and establishes disease gene relationships. So if we apply this Genet here, you'll see we get a much shorter list of genes, right? So whereas using the HPO, we're bringing in everything, being very sensitive, but sometimes getting too many results. This Genet actually has a more accurate set of results of genes that are likely to be involved in the phenotype we see. And this is because it's not just looking at each HPO term individually, it's looking at the groups of HPO terms and seeing which groups of HPO terms are commonly found in rare disease. So let's see if there's any de novos and any genes spotted by this Genet. Oh, and that surprised me slightly because I would swear it worked earlier today. Okay. Okay, so I'll have to prove that we get the result anyway. So we'll go back to the HPO. We run the query. We have 6 possibilities here. So we would I mean, as I say, in theory, I got a bit further than this earlier this morning, but I must have done something slightly different. Anyway, six variants would usually be enough that you can handle it manually, right? You can see which ones look most information here from most informative here from the color coding, but this color coding is just telling you how likely the variant is to have an impact on the gene. It doesn't mean that the gene is necessarily involved in disease, right? So my T1L, well, it is a good candidate because it's a myelin transcription factor 1-like, and it is autosomal dominant, right? But if you notice the phenotype here, the phenotype includes is mental retardation, and our child specifically does not have any intellectual disability. So you would go through these one by one, or if you know very well and you're the clinical researcher, you know, if you're an expert in neuromuscular diseases, you would know that RIRE1 is actually a very good candidate gene. And you wouldn't need to go to OMIM because you would already have this knowledge in your head. But RIRE1 is a code for ryanodine receptor. And you can see it's implicated in the autosomal, a few autosomal conditions here, but specifically in this case, in central core disease, which is a mini core myopathy. And indeed, in this case, it was confirmed that this was a de novo variant in this individual, a de novo variant in RIRE1 that that was resulting in a diagnosis also of central core disease. So they actually discovered 2 things here. So one, they got the actual disease because they weren't sure before. You know, it had from the HPO they had an idea of what it could be, but once they knew that it had this variant, the novel variant that was damaging the RIRE1 protein. Then they could confirm it was central core disease, and also they knew what the cause of the rare disease was that the child had. Okay, so sorry for the little glitch there near the end, but I think you've seen how the platform works. And now we can go to any questions we might have.
Thanks, Steve. Yeah, so we can start. There are a couple of questions. The first one is if there is any written explanation of these filters as well. I guess for the annotations, if there are, you're muted, Steve.
Sorry, Leslie. Did you see the pop-ups when I was doing the demo?
Yes, we saw it, but maybe it's good to.
Yeah. So we have links out, right, to all these things, right? So they all tell you where they come from. So let me just show you again, then, since you were seeing it. Okay, I'll share my desktop this time. Do you see the platform again?
Yes.
Okay. So for example, in the variant class, this is actually a link out to the ontology. So this will tell you what the meanings are of the different types of variants. ClinVar, obviously, I think everybody knows. We have tooltips here on the major categories as well. And we feel most of the information is easily discoverable. We do have some explanation, but not very detailed explanation of specifically what each resource is. So you would have to go and look as we're constantly adding resources. So it would be a challenge for us to maybe keep that up to date, but it's maybe something we should consider.
Thank you. There is another question concerning the allele frequency filters. Are these different for recessive versus dominant inheritance?
Well, you would want to apply a different value for recessive versus dominant inheritance, right? But the actual, the number you put in is the allele frequency that has been seen in the database, right? So 0.01 in Nomad means that 1% of all chromosomes in Nomad had that variant, right? So for a rare disease, if it's autosomal recessive, I used 0.02 because that's 1 in 50. And the probability of you getting 2 copies, if that's the population prevalence, the probability of an individual having 2 copies is 1 in 2,500, right? But if you know a disease is autosomal dominant, then you wouldn't be using 0.02 because that would be 1 in 50, right? Because they're just one allele. So if it's one allele that causes an autosomal dominant disease, you would want to go down to 1 in 10,000, for example. So you would be entering 0.0001 there.
Thanks, Steve. Then there are 2 questions that are related to the internal frequency. So can you explain a little bit if which samples are inside this internal frequency database, and if individuals are unaffected only or affected and unaffected, and from samples for exome or genomes?
Exactly. So the internal frequency contains the allele frequency that's observed across all individuals in RD-Connect GPAP, right? So we have about 20,000 individuals at the moment. So you're talking about 40,000 chromosomes. So if you'd seen once, it will be 1 over 40,000. If it's seen twice, it will be 1 in 20,000. So the allele frequency internally, the reason why we would apply it as a filter as well is because there are a few positions in the genome that are known to be difficult to map. So you get alleles at quite high frequencies there, and you would, depending on the pipeline, they may differ from our pipeline slightly from Nomad's. So you would want to remove those positions because they're going to be technical false positives. There's only a few, but occasionally it does help a little bit with the filtering. It saves you wasting time on something that would otherwise there's not going to be an explanation for your case.
Thanks. Then there is a question related if I have a candidate novel gene for a disorder, and I would like to find patients with compound heterozygous variants in the gene of interest. How can I do it? And if I can contact the researchers or physicians that own those patients. So I guess the question goes to, if I have a candidate gene, can I identify other patients with variants in that gene, and how can I contact them?
Yeah, so I didn't cover this today because this is the more advanced features that Leslie will cover in the next webinar in a couple of weeks' time. But to give a basic answer, yes, the answer is there's a couple of ways of doing this, right? So essentially what you're trying to do is what we call patient matchmaking. So you're trying to find a patient who looks like yours phenotypically, who has similar types of variants, because then those variants might be the explanation for the disease. So we have a couple of ways of doing this, and one way is you can search across all the samples for which you have access, and by gene you can see which variants are found in that gene and with which frequency in the platform, and then you can contact the submitter of any variants that you're particularly interested in. So an email will go to both of you and you will be put in contact. The second way we can do this is through matchmaking, as in matchmaking exchange. So matchmaking exchange is a network where if you have a case and you found some variants, but you're not sure if your variants are going to be causative, are likely to explain your, you have an idea that they may be the explanation for the phenotype of your case, but you're not sure. You can send these variants together with the phenotypic description, basically in the HPO terms. So you send the HPO terms and the variants out to other members of the Matchmaker Exchange network, and you ask them do you have a case that looks similar to this and has variants in this gene? And you get a response back with a score, and if successful match is made, you get a response back with a score, and if the score is high enough, then you probably want to try to get in contact with the submitter at the other end. And we facilitate that both within RD-Connect and with partners in Matchmaker Exchange.
Thank you, Steve. Then there is a question related to what happens once I submit my sample. So are these available to everybody else in RD-Connect? Is that a condition to use the platform? And in the same sense, if someone else can analyze any exome or genome uploaded in GPAP?
Yeah, so we very strongly believe, and this was, you know, one of the core beliefs in the RD-Connect project, that the way forward to help the rare disease community to get faster diagnosis is through sharing data. So the idea is that you share, you have permission, of course, you need to have permission to share your patient's data, and you share it in the system. So you have to put in the phenotypic information, the description, and then you send us the sequencing data, which can be FASTQs, BAMs, or CRAMs. What we do is we put it all through the same standard analysis pipeline, so the data is consistent throughout, so we don't have batch effects, and then it's uploaded to this platform. If this is brand new data that you've never looked at before, you can apply for an embargo of up to 6 months. So that means that only your team or any other groups within the platform that you actively share it with will have access to the data. After 6 months, the data then becomes visible within the platform. But by visible, I mean what I showed you today. So you cannot see other people's PhenoStore records, but what you can see is the diagram of the pedigree and the HPO terms. And you do have access to other experiments, right? So you can look for a similar type of experiment and perform analyses with your experiments and somebody else's data. But realistically, the most common way you would do that, again, is using what was alluded to before, the search across all, or some sort of matchmaking, whereby you do your own filtering, and then you look for cases that look like, you ask the database essentially, are there other cases that look like mine? Does that answer the full question?
I think so. And then another question is it possible to query on range of homozygosity in consanguineous families?
Yes. So again, this is a feature that Leslie might cover next time. So yes, one feature of the standard analysis pipeline is we identify all potential long runs of homozygosity over 500 kb in length, so half a megabase. And one filter that I didn't show is that you can filter directly to only show variants that follow their inheritance type that are in runs in the cases of over 500 kb, 1 megabase, or 2 megabases. And indeed, for consanguineous families, particularly if for example, the parents were cousins, then this will, in about 80% of the cases, that we would expect to find your causative variants in such a run.
Thanks, Steve. I was just thinking related to the previous question on accessing exomes and genome from other samples. When you register, you also sign a code of conduct, so no one can publish any result from your own samples without notifying you before, in case that this was also a concern.
Yeah, good point.
So next question is if we plan to introduce interpretation of CNVs, SVs in the GPAP.
So yes, we do. Well, SVs for genomes, potentially yes. For exomes, you can't really call structural variants. But yes, you might have seen in the demo there, we mentioned that we have a CNV. So we already have a prototype for how we would show copy number variant data in the platform. And the idea would be that you would be able to apply filters to both the copy number variant and the SNV and small indel variant data at the same time. For example, you might find a compound heterozygous case where in one allele you have a significantly impactful SNV, but the other allele you have a deletion or a duplication helping lead to the disease.
Thanks, Steve. Then quickly, the last two questions, I think. One is if the data need to be trios, or we analyze other relatives, for example, cousins or aunts affected?
No, we use trios in the example because trios are generally a very useful case, right? But you can upload singletons. There's no problem in just uploading the case. Obviously, if you upload the case alone, then you will get a longer, it will be harder for you to filter down to small numbers, right? You're going to get a longer list of variants, particularly if you're looking for autosomal dominant, because you get a long list of heterozygotes. But it's still possible, obviously, with the gene lists. It depends how well defined, how well defined your phenotype is. If you want to apply a very short gene list, then you'll be able to see if you have variants in those genes. So we take all pedigrees of all formats.
Thanks, Steve. Then there's a question that says, besides SolveRD, which other type of projects are eligible for submission of data to RD-Connect? So here, maybe you can explain a little bit who can register and which data is accepted.
So let me actually share again, because I have one slide on this, I think. So essentially, this is what you have to do to join RD-Connect, right? So there's an online registration. You go to platform.rdconnect.eu, look for user registration. We have an adherence agreement and a code of conduct, as mentioned by Leslie. So this is generally signed by the PI or the head of the group. And once we receive their application, we confirm who they are, and we actually send the application to a independent data access committee who confirms whether they agree these people are bona fide or not. So basically, you should be at a research institution or a hospital and interested in rare diseases, right? Then you submit your phenotypic data, your metadata, your sequencing data. We reprocess, upload, and make it available to you. So this is open to anybody who has the legal permission and wants to share their data within RD-Connect.
Yes, and I guess any project or any, you can submit either a few samples or data sets or be part of a bigger project. It's not a limitation in the
Exactly.
the platform, right?
So the large part of the data we have, or most of the data now, has come from SolveRD, obviously. But we have other submitters who are like a hospital who has an unusual case, and they've sequenced the child and the parents, or they somewhere, and they send us the data, and we upload it, and then they can analyze their own data. They analyze the variants themselves to hopefully diagnose their cases. So there's no sample size is too small.
Thanks, Steve. And really last one before I give back to Cornelia. So they ask if there is a consent form that has to be signed by the patients, or how is this managed?
Yeah, so no. So essentially the PI, when the PI is responsible for that, right? So the PI, when they assign the adherence agreement, they're saying for any samples that they do submit that they have correct consent, in this case for sharing for research purposes, right? So we don't have specific consent forms, and we don't check anybody's consent form. The obligation there would be on the PI who's responsible for submitting the data from his team.
Perfect. Thanks a lot, Steve. So Cornelia, I don't know if you want to close the session.
So yes, so now with this, we are at the end of today's webinar, and I would like to thank the speaker and the panel, and of course all of you for joining. And I would like to invite all of you to join also the second webinar of the series on the 19th of October. So the registration is still open, and I think we have the link also in the chat. So thank you all and goodbye.
Thank you, everybody. Bye-bye.